All right, sounds good. Thank you for the introduction. Um, so if you have any questions at any point, you can put them in the chat, but I think for the sake of time and so it doesn't disrupt my thoughts, I'll answer the questions at the end. But yeah, so today I'm going to talk about research, botanical research on lower Mississippi River islands. And so the first part of this presentation is going to cover a flora that I did with another lab mate, Grace McCartha, and then some other people who I will talk about that also did that flora. But just because I want to give a background about these islands before I talk about my research, and I'm not going to go too in depth to that flora research and the results because you can find a presentation on YouTube through the Arkansas Native Plant Society that Grace McCartha did on the flora, and then also some community assemblage research and results and everything like that. So I definitely recommend that you find that on the Arkansas Native Plant Society's YouTube page if you want to learn more. I can also answer questions about that as well at the end, but I'm not going to go too in depth about specific things about islands and communities and I don't know, more details about species finds, but just a little bit, just to make it interesting and some background. So just a little bit more about me. I am from the St. Louis region, so I'm actually from Old Monroe, Missouri, which is kind of like 45 minutes, a little bit north of St. Louis. And as you can see, it's this red point here. It's still pretty close to the Mississippi River, so I've grown up next to the river dealing with flooding from the river and basement floods and road closures. So I've grown up near the Mississippi River my entire life, but haven't spent too much time actually on the river up until graduate school. And so I did my undergraduate at Southeast Missouri State University, and I majored in wildlife and conservation. And while I was an undergraduate student, I was really interested in big cats and canids. So I volunteered at Crown Ridge Tiger Sanctuary. And then in the summer, I did an internship at the Endangered Wolf Center in St. Louis. And that's what this picture here in the top middle is, me doing a paw painting with one of the maned wolves there. And I realized after doing that volunteering and that internship that I wasn't really that interested in animals like I thought I was. And I was definitely not interested in zookeeping or being a keeper at all after doing those jobs. It just wasn't for me. And I'm more interested in plants. And I took a few field botany courses out as well while I was at SEMO. And that's where I really started getting really interested in native plants and going out and looking at plants and botanizing. And then after I graduated in 2019, I interned at the Litzinger Road Ecology Center, just a summer internship. And as part of that internship, during the summer, you have to complete kind of a research project on site. And so I decided to survey the algal diversity that was on the creek there. And so that's what this bottom middle picture is. It's a Platydorina, it's in the genus Platydorina, and it's just a green algae that I found. And that was really cool. I was really interested in algae at that time. I just thought it was fun to look under the microscope and look at pond water, or I guess creek water in this situation. And I have not done anything with algae since then. That was just a kind of a one-time thing. It was really hard to key out algae, even to genus, but it was really fun and interesting. And so after I completed that internship, and then a few months passed, I started graduate school at Arkansas State University to start my master's, and I joined Travis Marsico's lab, which this picture here on the top right is my lab group, and this guy here in the blue shirt, that's Travis Marsico. He's a really great person, and I love being in his lab, and yeah, he's just been a great advisor. And so these are my other lab mates, and we took that picture while we were at while we were at the International Biogeography Society in Vancouver last June. And so I am still technically a student. I don't graduate till May, but I just defended my thesis in December successfully. And so kind of missed the cutoff for that with how things work out. And so I'll be graduating in May, but luckily I was able to start my full-time position back up in St. Louis, kind of where I'm from. And it's actually at Litzinger Road Ecology Center. So it's the same place that I interned at. And so this is a division of the Missouri Botanical Garden, but it's not open to the public. So it's private, and it's just kind of for place-based learning and for school groups to come out and teachers to learn how they can incorporate teaching about native plants and ecology and restoration and things like that to their students. But I work for the restoration side, and I am the restoration coordinator for Project, which is an area that has been cleared out to put a big sewer pipeline in. And so over the next 4 years, I'll be there kind of starting the restoration for that. And then also I have a cat. Her name is Jackie, pictured there. I adopted her while I lived in Arkansas, and her name is Jackie, and I named her after a lower Mississippi River island, Jackson Point. Okay, so enough about me. I'm going to talk about the Mississippi River. So the Mississippi River is the largest river basin in North America. It's the third largest river basin in the world, right behind the Amazon and Congo river basins. It covers a vast majority of the contiguous United States and drains about 40% of the lower United States as well. And it's divided into 3 parts. So the upper Mississippi River starts at its origin in Lake Itasca, Minnesota. And then that ends at the confluence of the Missouri River and the Mississippi. And then the middle Mississippi River is from the Missouri to the Ohio. And then the lower Mississippi River is from the Ohio River, kind of near Cairo, Illinois, down all the way till, to the Gulf of Mexico, where it empties into there. So this research, all research that I'm going to talk about today and that we focused on in our lab was completed on the lower Mississippi River. And so the lower Mississippi River is a little bit different than the upper and middle Mississippi rivers in that it's in the Mississippi alluvial plain. And so it's a really flat area. And so the river has naturally meandered and kind of cut through the soft sediment of that area. And so you can see evidence of that through these old river chutes and oxbows. And so that kind of helps create islands as well. And then, so these islands are kind of formed from that. Sometimes they're formed from sedimentation following wing dikes. And then sometimes steamboat wrecks as well. And so right now, the Army Corps of Engineers does all the managing for the Mississippi River and the lower Mississippi River. And so they'll put in things like these wing dikes that are pictured here. They'll put up bank revetments, and they'll do channeling and dredging to keep the channel deep for transportation, barge transportation. And so river does not move around and meander like it used to. So it's heavily modified now, and I'm not sure that many islands will form in the future. I don't know, that would be interesting. And so the Mississippi River also follows a pattern. And so typically, although it varies year by year, the river level is higher in the late winter and early spring. And so this hydrograph here just shows the river stage for 2021 from a nearby river gauge near Helena or Greenville, Mississippi. And then the green line is just the historic average. So as you can see, it's usually highest in the late winter and spring, and then lowest again in the late summer and fall. And so what that means is that the islands on the lower Mississippi River experience a flooding regime in which the higher elevations are flooded for very little or not at all. Just they're not flooded as much as the lower elevations. And so at those lower elevations, the plants can't grow there because the elevation is underwater. The soil is waterlogged, and then there's the current from the river, which is pretty intuitive and obvious, but really plays a big part into Grace and I's research with elevation. And so globally, flooding is expected to increase with precipitation overall increasing, and especially so we're going to see changes, or scientists are expecting to see changes on the lower Mississippi River just because of this increased precipitation coinciding with earlier snowmelt from the north, and then that all emptying into the Mississippi River. And so the flooding is going to be greater in the future. And so we don't really know how plants and the ecosystem will respond to this change in the flooding regime, but it is probable that there will be some alterations to these habitats and ecosystems. So getting into the flora now. So I completed this flora with Grace McCarthy. So we both started in Travis's Travis Marsico's lab at the same time because he wanted to recruit 2 graduate students, because going out on the Mississippi River is kind of hard and can be dangerous. And so it's safer to go out with 2 people, and we both were interested in these islands, and so we were going to work together on them. And so Grace and I completed this flora, which is a vascular plant inventory of 6 lower Mississippi River islands. And Grace created this really cool map. And so our most northern island is Buck Island, and that is about 45 minutes south of Memphis, Tennessee, and then which is about 2 hours driving south of Jonesboro, which is where Arkansas State University is. And then our most southern island is in Greenville, Mississippi. So that's about a 4-hour drive south of where we were at school. So we did a lot of driving for this research, but we kind of just picked islands. Grace had this aspect to a project where she wanted to see if the species compositions would be different above and below those major river inputs, so the Arkansas and White rivers. And so we kind of selected islands based on that. And then also these islands are mostly privately owned for hunting clubs, but 2 of the islands, Buck Island and Montezuma Island, or not Montezuma, sorry, Choctaw Bar Island, are owned and managed by the Arkansas Game and Fish. So you can go there and hunt, and then also you can go out to those islands and use them for recreation, hike around, or just sit on the beach. And so we completed this flora during the summer and fall of 2020, and then spring of 2021, just so we could get all of the different seasonal flora. And then we would just go to the island and do meandering surveys. And so sometimes we would look at, or usually we would look at Google Earth and look at satellite imagery and kind of look for cool areas around the island. Like that looks like a sand dune area, so we want to go there and see if there's cool species, or we can see a slough here, so we should boat here. But sometimes it was about accessibility. And so if we saw an area on the island that looked accessible for us to boat up to, sometimes that's just kind of how we picked our starting point. But we always tried to get the top of the island, middle, and then the southern portion. But as you can see, these red dots on these island maps, those are our tracks. So that's actually where we were on the island. And you can see some of these islands we missed a lot, and they're pretty big islands. So, and this was multiple day trips. It was just hard to get the entire island. And so when we would get to an island, we would collect every species that we encountered. So if we saw a cottonwood on all 6 islands, we collected it 6 times, so we'd have a voucher from each island. And we often collected duplicates as well, because we received funding from Arkansas Natural Heritage Commission to complete this flora. And then during the time of collection, we would take a picture for iNaturalist so that we could associate it on our herbarium annotation label and use it for identification, so that we have a press specimen, and then on the label you can scan a QR code and see our iNaturalist picture as well. And all of those iNaturalist observations are available still, so if you were ever interested, you can find these projects on iNaturalist and see all of the cool flora that we found. And so every herbarium specimen was deposited in either STAR, which is Arkansas State University's herbarium, or Arkansas Natural Heritage Herbarium. So between Grace and I, we collected 2,310 specimens, which is 485 species out of those about 2,000. And then 491 taxa, which includes infraspecific taxa, so things like subspecies and varieties. There was 89 plant families, and then the most species-rich families were Poaceae, Asteraceae, and Cyperaceae, which also are the most speciose families in North America. And then 390 are native and 101 introduced, but only 29 were considered invasive. And we found 5 Arkansas state records and 7 Mississippi state records. So that just means that those species that we collected were not officially documented from the state yet. And then we found 266 total county records. And yeah, this top picture here is just Grace and I. We found grass. I believe this is a Synchris species. I can't remember its common name, but it's a spiky grass that gets stuck in your shoe when you walk on it. And so this is kind of what it looked like for us. We would find something and find a plant, dig it up out of the ground, and then take a picture and then put it in our nifty little Ziploc bags that we had uniquely numbered so we could keep track of what species were in what bag. And that way we could walk around all day on the island and have our plants with us. And then the bottom picture, you can really see we were at the tip of the vegetated area of Choctaw Island. And here's Travis, our advisor, Grace, and then me. And we're just kind of looking at how vast the sandbar extends beyond the island. It's pretty cool. So here are our 12 state records. So some are state records in Mississippi, some are in Arkansas, but Equisetum variegatum. And so the pictures also kind of line up with how the BONAP map is laid out. So Equisetum variegatum is here in the top left, and then Acaranthes japonica, Euploca convolvulacea. I'm sorry that I don't know a lot of common names, but feel free to look them up, take a picture or something. That's just information. It's hard enough to remember scientific names. I'm sure you all can understand. And so remembering a common name is hard as well. And so Euploca convolvulacea was a cool find because you can see, I know the BONAP is really small, but you can see it's like traveling down the Arkansas River. So it was a species range extension. And then same with this Acaranthes japonica. It's like Japanese chaff flower or something like that, following the Ohio River down to the Missouri. Yeah, so really cool things that you can see how they really are traveling down the river. Cicimbrium locellii, Stellaria aquatica, Chyturus meriviastrum, Mentha canadensis. I'm sorry, this picture is just really bad, but it's from my iNaturalist. Just a mint. And then Potentilla norvegica and Potentilla paradoxa, so 2 strawberries, a ground cherry, Physalis mollis, and then Cyperus diandrus, so flat sedge. So yeah, these were really exciting. Of course, we didn't know at the time of collection that we had found a state record. It was always whenever we would finally get to identifying our collections that we would figure out that they were state records. And so every specimen was identified by either me, Grace, or another graduate student who eventually jumped in on this project, Brendan Kosnick. He kind of helped, or he helped with the spring flora and then also a lot of plant identification as well. So we would sit in the herbarium and just run through the key for all of these species, even if it was something like cottonwood or sycamore. We still just wanted to run it through the key and learn the details and the features and characteristics that make it what it is. So it's really interesting. And so now these are just pretty flower pictures. I can't say that these were everyone's favorites, but I know that these were favorites, not just mine, but we would get excited when we find these in flower during the flora. So American groundnut, really cute. Toothache tree, this was a fun one to chew on and make your mouth go numb. And then anglepod, so milkweed vine, the monkey flower, Mimulus ringens, which I believe this is a trapped species in Arkansas. Physalis mollis, so ground cherries. There were different species of ground cherries that Grace and I really liked to look at and take pictures. I think those were both her and I. Those were our favorites for sure. And then bird cucumbers, so Sicuus ungulatus, really cool, cute flowers and cool fruits. And then Melanthera nivea. So this is an Asteraceae, a really cool flower and plant. And then dodder is really cute, but it's even cuter when it's in flower. So just to talk a little bit about the habitats that we found on these islands, there were a lot of cool and interesting habitats. I'm not going to talk about all of them, but I'm going to talk about the most common ones that we would encounter. So a lot of islands had these willow sandbars or these willow forests. So it was black willow dominating the canopy. It's really cool. Really pretty photos. And then sometimes it would be bare with just sand. And then sometimes there would be ground cover like this looks like a lot of morning glory vine. And then there would be Mesic mudflat forests. And so these would be kind of at the higher elevations, the areas that didn't get flooded a lot. And so this is where we would find Carex grayi and then Lobelia cardinalis, so the cardinal flower, lots of sedges or carices. There'd be lots of flat sedges on the beaches and weedy little things as well. And then lots of violets, like the dominant ground cover sometimes would be violets in those higher Mesic forests. And then vines were really common on the island. So this left picture is Grace standing next to a giant grape liana. Those were so cool. We really liked those. And then just grapes covering everything. And then this was our last trip out to the islands to do plot work for Grace's community assemblage research. And so we were just kind of taking it in, this which would be a salix or willow forest, but was covered in the bird cucumber just all over. It's just like a monoculture of bird cucumber, and it was really cool and really pretty. And then yeah, Brendan and Matthew, the undergraduate student in our lab, were just kind of looking at it, and it was really cool. And sometimes there would be ephemeral swamps on some islands and sloughs, and those swamps would have kind of a soup of duckweed and floating liverworts. Another cool thing that we found in a slough on one of the islands was this water clover fern, Marsilia vestita, and this is tracked in Mississippi, so it was a cool find. And then on some of the larger islands, there would be these shrub savanna type habitats where there was mostly herbaceous kind of grassland type, almost looked like a golf course sometimes, and it would be mostly Bermuda grass. I think they planted Bermuda grass for hunting. These would be the privately owned hunting club islands that would have these. And then these stands of cottonwoods with really unique things and cool plants growing underneath them. And then the last habitat type, which was all of our favorite, I think, was the grassy sand dune. And this is where you could find tumbleweed, so Cycloloma atriplycifolia, and just really cool. And some grasses and other pretty asteraceous flowers. We'd find something like Osage orange here. really cool habitat. And as you can see, it's just really pretty. It's like grass scattered throughout and then sand scattered and sometimes tumbleweeds here. It's so cool, so pretty, and really easy to walk through. So this first year that Grace and I went out to do this flora was for us to get just a baseline of what species grew on the islands and just like what these islands looked like, because we really didn't know. And our advisor Travis didn't really know. So that was kind of the goal of the flora, and to help us come up with questions for our own research. And a really huge observation that we noticed with these islands was how great the elevation difference is. And you can really see that here in this picture on the left and the right. So Grace and Travis are standing here, and they look so small compared to the rest of the island, which has a huge elevation gradient, sometimes like 30 feet we'd have to climb. And then here again, this is on our most southern island, Lakeport. And we walked all the way up here, and Grace remembered that she forgot something at the boat. So she had to walk all the way down and over these rocks, and I'm standing here just kind of watching her walk back up. And another thing that I should mention, yeah, sorry. If you're wondering who's taking these pictures, is Richard Abbott, who I know gave a talk recently. So he is going to be a co-author on this phenology research. He's a co-author on the flora, which we're in the review stage now. So we've kind of got that submitted and kind of working on that now. But he helped us a lot with field work and plant identification in the field. He went out with Grace and I almost every time we would go out to the islands. He really loved the field work. We spent a lot of time with him, and he taught Grace and I pretty much what we know now for how to identify plants. And so, yeah, we couldn't have identified as many plants or done this research without him. He was a lot of help and gave us a lot of advice, and more than just this plant is this plant because I say it is. He would tell us the features and the motifs that he likes to call them. And yeah, he was a lot of help, and it was really fun working with him. So, of course, we saw other things that weren't just plants, even though we were there to look for plants. So we saw some Animal Crossing. I don't know if anyone plays Animal Crossing on Nintendo at all, but I do. So I thought I would put that little thing in here. So yeah, we'd see some pretty neat stuff. We would see deer and feral hogs. We never saw the feral hogs, but we'd see their tracks all over. We'd see deer. We've seen coyote. We've seen black bear. So those are some really cool things that we didn't take pictures of. And just neat little animal tracks on the beach. I don't know what animal left these, but they were really cute. If anyone knows, feel free to put it in the chat. Maybe a rabbit or a turtle. Although I don't remember seeing rabbits. We see beavers. So green snakes, sphinx moth, this really pretty green iridescent weevil, pipevine swallowtail, Io moth, and then box turtles. There were a lot of box turtles. I think that was our most commonly observed turtle species on the islands. So yeah, it was really fun. A lot of times we would scare the animals away before we could see them because we'd be loud and talking and botanizing through the woods, but sometimes we get to see some really cool things. And then of course there's lots of birds, least terns and killdeer, pelicans, seagulls, kingfishers, herons. Yeah, lots of cool stuff. These islands certainly have a lot of wildlife, and they're great places for animals to go that are pretty much untouched for the most part. And then just to kind of end this little flora section, it was a really good experience, but also really hard field work. I'm not sure I'll do field work as hard as this ever again, just because I don't know if anything could beat it. I would be totally down to do this research again, though, and experience it all again. But it is hard to do field work on these islands because you have to drive the truck with the boat to the boat ramp and then make sure, hoping that the boat is working. We had a lot of boat issues where we'd get down to the boat ramp and suddenly our boat wouldn't work, so we'd have to troubleshoot. So that was always iffy and kind of frustrating to deal with. So that was a whole hurdle in itself. And then actually getting out onto the river is really dangerous. There's eddies and whirlpools and wing dikes and large trees that go down the river. And then also, as you can see in this bottom picture here, large barges. So they create a lot of waves. You have to be really careful around them and make sure you don't get too close to them and give them their space. And you can see here, this is our little boat compared to this barge. So yeah, very, very small and really dangerous. We're very remote. You have to take a boat to get to the boat ramp, put your boat onto the truck, and then be able to go somewhere. So we had to be careful and try not to get hurt. But of course, sometimes there would still be biting and stinging insects. And so we all had to deal with a lot of yellow jackets and a lot of mosquitoes and ticks. And yeah, so the usual fieldwork troubles, but it felt amplified by having the barrier of the river between us and the rest of the world. Okay, so I'm going to talk a little bit now about my thesis research, since I gave you that background on Mississippi River Islands. Again, if you want more information, you can ask questions or at the end, or find Grace's Arkansas Native Plant Society presentation. So my thesis research had two components, and they were phenology based. So I focused on plant reproductive phenology. And so I had a field study, which was on Buck Island, a Mississippi River island. And I just wanted to determine if river island elevation, which is associated with inundation and how long each elevation would be flooded, was significantly influenced by significantly influenced phenology and plant fecundity, which plant fecundity is the ability to produce offspring. So I kind of had this field component, seeing if flooding altered plant phenology. And then I had an herbarium specimen study, so just looking at changes in phenology over a larger spatial scale, so lots of or a large latitudinal range, but I'm not going to include those results in this presentation. And then, so getting into phenology. So phenology is the timing of events and seasonal development of an organism. And so for my project, I focus on plant reproductive phenology, which is sensitive to environmental and climatic factors. And because of that, it is often used as an indicator of climate change. So advanced flowering and fruiting onset has been widely documented in temperate systems as signs of global climate change become more prominent. And so these studies are focusing on how phenology might be advancing based on climatic factors. And so there are lots of studies in temperate Asia and then temperate Europe and also temperate North America. So that's something that is really commonly studied with respect to phenology. And so what is not so much researched, as far as I have seen and done a lot of researching and looking at the literature, but how flooding might be affecting plants in temperate systems. So a lot of research is focusing on climatic factors in temperate systems. And so I wanted to kind of fill this gap or just learn more about how flooding would alter these areas, since our flooding is so dependent on the seasons and in the temperate system. And so that's not to say that there isn't phenological research completed with inundation, but it's done either in experimental settings, like in a greenhouse or something, or in tropical habitats on tropical species where precipitation might be impacting phenology more than temperature here. And so again, this study occurred on the lower Mississippi River on Buck Island, which is the most northern island of the 6 that we used in the flora. And here on the left, you can see this is just kind of a snippet of the lower Mississippi River Island. This area is surrounded by land that's used for row crop agriculture. And you can see that in the light green areas, and then the dark green areas are the remaining forested areas. And then here on the right, this is just Buck Island, more zoomed in satellite imagery, and it is located right outside Helena, West Helena, Arkansas. So Buck Island is owned and managed by the Arkansas Game and Fish Commission. It has a variety of habitats. Some I've already kind of talked about. So it has those higher elevation mesic forests. It has willow woodlands. It also has a swamp in the middle of it. And then it also has sandy and muddy banks where a lot of weedy species occur. And that's also primarily where I did a lot of monitoring for phenology. And so this island is about 3 miles long, and it has an area of 686 hectares. It's about 150 years old, and it actually has a pretty tragic origin. It started forming after sedimentation from a steamboat wreck, and the steamboat was named the Niagara, and it had a collision with another boat in 1865, and it had 150 passengers on it, and unfortunately all of them passed away from that wreck. So pretty tragic origin, but also kind of interesting in the way that islands can form following sedimentation like that. Right now it's used for recreation and then public hunting land as well. So for this project, just 2 of my main predictions that I had. So I had the first one was that plants at lower elevations would have reproductive times that are later in the year compared to plants at higher elevations. And then the second one was that plants at higher elevations will produce more fruits than plants at lower elevations, so they'll have higher fecundity. And that's because of the longer reproductive season at those higher elevations. So I tracked 6 taxa, and so they're all native and kind of commonly found throughout the island. They're kind of weedy and would be able to find them throughout the elevation gradient. So Maluga verticillata, and then Roripa palustris and Roripa sessiliflora. They're both native and they have similar and overlapping phenology. So I just grouped them as one group because I couldn't identify them until they became fertile. So I knew already that they would have overlapping phenology. So they're just 2 species as one for analyzations. And then Solanum emulans, oxalostricta, Phyllolanceolata, and then Violosauroria. So we monitored 6 herbaceous, those 6 herbaceous species from April through December 2021. So it was really long field season. And so during each visit, we would count the number of structures in a phenophase. So like how many flower buds, how many open flowers, developing fruits, and then mature fruits. And so each individually tagged plant would be counted during each visit. And that's here in this bottom right, you can see what the tags with their unique numbers looked like. And so we'd visit twice a week in the summer and then once a week in the fall. And we would see the same plants every visit. So during this whole time, we established 62 habitat patches that ranged in elevation from 46 to 57 meters. And each habitat patch would have 2 to 10 individuals in it. And you can see the location of these habitat patches here on the island. And then this is just kind of showing you how big that elevation gradient was on Buck Island. And then here, these pink flags are what a habitat patch would look like from the different distance, just so we could see it. And as you might expect, the lower island elevations were more frequently inundated. And sometimes that would happen after we would establish a habitat patch. And that's what this bottom picture here is showing. So we had established this patch and began monitoring it. And then suddenly, I guess there was a lot of rain up north and we had a lot of flooding. And you can see that line up here. This is how high that water went. And so when that would happen, we would collect the tags and not reuse them and just wait for new plants to grow on those elevations so that we could reestablish new patches and start monitoring phenology again. And so that's what that picture of Grace there, she's picking up patches or flags after flooding. And then another fun component of this research was how much or how often I would have to track the river gauge. It felt like daily I was looking at the river gauge to see how high the river would be so that I would know if we would even be able to get out to the island and see the habitat patches. Luckily, we were always able to get to the island, but sometimes if the water was below 8 feet, we would have to paddle instead of using our motor. So we knew it would be a longer field day, which was hard. But we weren't able to use the motor because we would end up sucking a bunch of sand and mud into our engine, which would clog the engine and then make it overheat because it uses water to cool the engine down. So that was very frustrating to deal with sometimes. And that's what this picture on the right here is, Travis and Brendan. And so we had to pull the boat through. We went to a shallow area. We thought it was deeper than it was. We realized it wasn't so deep. And then we had to get out and walk it. So we dealt with a lot of things like that on the river. And so during this phenological monitoring season, the river fluctuated a lot. So it fluctuated from 44 to 53 meters in elevation. And so that's what this figure here shows, just 2021 river height and then all 62 of my habitat patches. And that doesn't mean that they were all established during this whole time, but it's just the elevation of the habitat patch. So you can see that the lower habitat patches, that elevation would have been inundated as much as 6 times. And then the higher habitat patches were sometimes not even, were not inundated. That elevation area wasn't. So there's a lot of fluctuations throughout that year. And then some habitat patches were just completely lost to flooding. And so, yeah, again, just have to start over when that would happen. So now a little bit of the results. Yeah, so there's a lot of conflicting results that I'll talk about in a few slides. But so this figure just shows the number of reproductive days per patch, so just how long the reproductive duration was. And so I thought that higher elevations would have a longer reproductive season. All species would be like that. But it turns out that wasn't the case, and it was only significant. There was only a significant relationship for Mulugo and Phyla. So that's kind of what the star there shows. And the other species, although they're plotted, they did not have a significant value. So really just Mulugo and Phyla had a longer reproductive duration at higher elevations than at lower elevations. Which would make sense because lower elevations were more inundated than the higher elevations. So at least those two species followed what I would have expected. And then as for fecundity, so the number of mature fruits increased with elevation, but only for 3 species. So again, Mulugo and Phyla, which would make sense since they have longer reproductive periods at higher elevation. You would expect them to be able to produce more fruits. But Raripa was not significant in the previous slide, but maybe something with its indeterminate inflorescence, so it can produce as many fruits and flowers as it can, since it's in the Brassicaceae, until environmental conditions are not favorable. So maybe those higher elevations just had more favorable conditions because of less flooding, and so it was able to produce a lot of fruits because of that cool Brassicaceae adaptation and thing that has going on. And then the last analysis that I'm going to share, I did do analyses on onset, peak, and cessation. So the start, the peak, and then the end of the phenological season, but I'm only going to share the peak just for the sake of time. And so plants at higher elevations had an earlier peak mature fruiting day of year than plants at lower elevations, but only for Malugo and Solanum. And then those two species also had earlier onset and cessation days of year at higher elevations as well. So they were the only two species that were completely shifted in phenology going down elevation. So that was an interesting find. They also had the largest elevation range of all taxa as well. And then I only use the mature fruiting phenophase for analyses, just because I felt like I had the biology of these species is kind of weird. So I was either going to use open flowers or mature fruits, and I chose mature fruits because with a lot of these species, or I think all of them, they would only have like 2 or 3, or depending relative to how big the plant was, not a lot of open flowers. And I feel like those two were the most biologically meaningful phenophases. And so I ended up just going with mature fruiting to show these shifts in phenology following elevation. So this graph is kind of a lot, or this table is kind of a lot, but I just wanted to show that only 2 species, so Malugo, the top row here, and then Solanum, they were the only species that had phenology completely shifted following elevation. And then it was kind of mixed. So Viola did not have any response to elevation, probably because it wasn't found at lower elevations and was really habitat specific to higher elevations, undisturbed areas. And then, yeah, Oxalis, Phyla, Raripa, they were not, they didn't have any shifts as far as their mature fruit peak and cessation. And then Oxalis was a weird one. It was only significant for its mature fruiting onset, and it actually behaved opposite than what I predicted, where higher elevations started producing more mature fruit later in the year compared to lower elevations. So that was kind of an interesting thing. And then again, my predictions were sort of partially supported, but not totally. And so some species kind of had results that were what I expected, and some species did not. And then only Solanum and Maluga had completely shifted phenology on that elevation gradient. And so that just shows that each of these species has their own specific needs and their own specific biology that they have adapted to. And so you can't make these generalizations for all of the species. And then just some interesting observations. So Oxalis, we all found out that it has a really cool way to disperse its seeds. So that video there is just showing the seeds kind of popping out as soon as you would touch this capsule. It was really cool. Sometimes you get hit in the eye. And then another interesting observation was that plants at lower elevations would generally be shorter than plants at higher elevations, but they would still produce a lot of fruits and flowers and everything. So it's like they were compensating for starting reproducing so late in the year by growing really short. And this was not just the species that I tracked, but many other species in the flora. It was something we noticed while we were just walking around too. It was really interesting. And then just some more fieldwork fun that we had doing this phenology monitoring. So we'd have to go out in the rain or heat and just kind of had to deal with it and go out there. We would be in mosquito suits sometimes. We'd be sitting in the sun, feeling like we were being cooked. Sometimes the boat would break down and we'd have to paddle back. That's what this picture at the bottom left is. And then, of course, on the Mississippi River, you have to deal with flying Asian carp. Sorry if that video is really loud. But yeah, that was always fun to worry about and think about going out on the river. So yeah, there was a lot of people that went into all of this research. And this is kind of set up mostly for my research, but also the flora, there were a lot of help, and we used our naturalists and all the landowners, and there was a lot of communication with them. But I'd really like to thank my advisor, Travis Marsico, and committee members, so Richard Abbott and Scott Mangan, and then also my lab mates and Marsico lab members for all of their help and input and fieldwork help. And then also Johnny Waldrop, who is the manager at Buck Island Wildlife Management Area. He was a lot of help as well. And then John Rusky, who is works for the Quapaw Canoe Company. That's a really cool company that goes out to river islands and tours. And so he gave us a lot of advice as well. And then I also was funded for this research as well. And so I'm really grateful for having the opportunity to do this. And so, sorry if that went a little longer, but I'll take any questions. But I do want to just kind of say this quote. I found this on a bench at a local restaurant in Helena, Arkansas, which I think they're closed now, but I thought the quote was really, really nice. And I don't know if any of you have spent time in this area, but it's a great area. And so this quote says, "This delta is a powerful land. It just leaps out at you. It seeps deep within you. The muddy Mississippi water leaves a stain on the soul that is virtually impossible to get out, assuming any fool would try." And then this picture is just Grace and I on our last day of field work on an island, and we were hugging it goodbye because we were going to miss it so much. So yeah, if you have any questions, I'll take them now.